Hi, welcome back. In today's video, well, I keep saying today, in this video, things to be looking for are the question of what is the central dogma of genetics? So this is something that you should come into the course being extremely familiar with. If you're not, then really take some time to seek out some resources. Um, you can find information in the PDFs that I've provided. There's some really excellent YouTube videos, but I think maybe when I touch on that in this lecture, it'll come back to you. So what is the central dogma of genetics? Next is, do all genes encode proteins, and do all parts of a gene encode their product? So whatever that product is, do all the parts of a gene actually go into encoding that? And then DNA is double-stranded. Is there one strand that encodes genes and the other strand doesn't encode genes? Is there a mixture? Some parts of each strand encode genes. What's the deal with that? Okay, so those are the things to kind of be looking for as we move through our lecture today. So I'm going to disappear myself now so you can focus on the screen. Maybe. Okay. Okay, so we're going to see DNA depicted a whole bunch of different ways in different lectures. So this one hopefully looks familiar from when we talked about the components of a nucleotide. So once again, we're looking at DNA. At the top, you can see 5' on the left, 3' on the right. And remember that that refers to the directionality of that deoxyribose sugar. So is the 5' carbon sticking up, or is the 3' carbon sticking up? Another important thing to remind you of that you're probably super familiar with, but it's going to be really important, is base pairing. So remember, A always pairs with T, G always pairs with C, adenine always pairs with thymine, guanine always pairs with cytosine. The adenine, the A paired with the T, these together are a base pair. So an A paired with a T is one base pair. Okay. A G paired with a C is one base pair. So it's a pair of bases. Okay. A couple of orienting things reminding you of some things. Okay, so in the human nuclear genome, and we're not just going to focus on humans the whole time, but this is a good place to start. In the human nuclear genome, there are 3.2 billion base pairs. So in your genome, there are 3.2 billion of these base pairs, and we're going to come back to that in the next video. For now, I'm just going to point out to you, as I said at the beginning, you're going to see DNA depicted a whole heck of a lot of different ways. Okay, so you're going to see DNA depicted, as I said before, where it shows the nucleotides like this. Okay, you're going to see DNA depicted as just a helix like this, maybe with letters indicating the bases. Okay, so the bases are adenine, thymine, guanine, cytosine. You might see it laid flat out like this, where it's just showing the letters. You might see you might see it laid flat out with both strands with just colors representing the bases. You're going to see arrows that indicate directionality, thick and thin segments that indicate different things. You'll see sometimes boxes, and then sometimes just a line. So all of these different things are representing the same molecule, okay? All of them are representing DNA. They're just emphasizing different components, different parts of DNA, depending on what we want to focus on. Okay? So just keep that in mind as we're moving through the course. You're going to see it depicted a whole lot of different ways. The type of depiction is generally based on what we're trying to emphasize, or what component, what characteristic of DNA we're aiming to highlight. So for this image, this slide, we're just going to depict DNA as a white line. So that thick white line, kind of in the middle of your screen, is DNA, is a strand of DNA. This is one single molecule of DNA. So this is one molecule of DNA. And it is 58 million base pairs long. Just this one molecule. This is one continuous strand of DNA. So this is an actual representation of a real section of human DNA, a section of your DNA. You have a molecule in your body of DNA that is 58 plus million base pairs long. Cool. Isn't that cool? Don't worry, we're going to come back to that and I'm going to explain that. Okay, so that's cool. And we are going to come back to that. We're going to zoom back out, but for right now, let's zoom in to a smaller section. Okay, let's zoom into a smaller section that's a mere 180,000 base pairs long. This section that is this mere 180,000 base pairs long is a gene, or this is representing a gene. Okay, this image is representing a gene. So let's first, we hear the word gene, we hear, I mean, it's just common in our language, right? Oh, it's in their genes. Oh, it's genetics. But we need to actually stop for a second and think and talk about what a gene is, because this is the basis of everything we're going to do in this course. So a gene is a section of DNA that is required for the synthesis of a functional protein or RNA. So it's a section of DNA that's required for the synthesis of a functional protein or RNA. So understanding this takes us to the central dogma of genetics. Okay, so this may look familiar to you. The central dogma of genetics. The central dogma of genetics is that we go from DNA to RNA to protein. This is the direction. It's one direction. Okay, this is hopefully super, super familiar. So DNA is transcribed into RNA, then RNA is translated into polypeptides, which are the primary structure of proteins. They have to get folded to actually be a functional protein. Hopefully you remember that from physiology. But the central dogma of genetics is DNA is transcribed to RNA, and then mRNA is translated to protein. Okay, so something you may have noticed there is that I said DNA is transcribed to RNA, and then mRNA is translated to protein. And that's really, really key, because this central dogma of genetics, which you've learned, it's correct. It's just incomplete. Okay, if what we're thinking about is what is a gene. Okay, so the definition you probably learned is that a section of DNA is a gene, is a section of DNA that is required for the synthesis of a functional protein. So you probably learned a very heavy focus on protein. Okay, DNA, RNA, protein. This isn't incorrect, it's just incomplete. Because the RNA that gets translated to protein is specifically mRNA. So you guys may remember we talked about different types of RNA. So only mRNA gets translated to protein. There are a whole host, and I've just listed a few here, of other types of RNA that aren't translated. So they don't get translated into protein, but they do really, really, really important stuff in the cell. We have to have them. Have to have them. Would be dead without them. So there are lots of types of RNA, and we're discovering more all the time. We're discovering more about our genome all the time that do important cellular work, do things that are critical to our cells surviving and therefore us surviving. But they do their work as an RNA. They aren't then translated into a protein. So that is why the current definition of a gene is it's a section of DNA that is required for the synthesis of a functional protein or RNA, because we have all of these RNAs that are doing important cellular work, okay? So it's not just a protein. Most of the genes that we know about, most of the genes that we're aware of are translated into a protein, but we're discovering more and more RNAs all the time that are doing cellular work. Okay, so that's the central dogma of genetics and our extension of what you probably learned as far as the definition of a gene. So now let's go back and let's look at our gene. So we're going back and we're looking at our gene, and our gene happens to be a gene called INSR, or insulin receptor. And this particular gene, like most of the genes that we are aware of, is transcribed into mRNA and then translated into a polypeptide, which is folded into a protein. As you may guess, insulin receptor, this protein is actually an insulin receptor. So it's embedded into the cell membrane, and it's a receptor for insulin, like you learned about hopefully in physiology. So I don't know if you guys, like, I think that's so cool. Okay, so this particular gene, our gene that we've been focusing on, that we've been using as an example to understand what a gene is, does encode a protein, as most of the ones we're aware of do. But you may have noticed that if we look at our gene, my little cartoon depiction of our gene, this pink line, that there are parts of this pink line that aren't exactly the same color, right? So there are different colors, and these represent different parts of a gene. So before we can get to our mRNA and our protein, we have to talk about those different parts of a gene. Okay, so these little slices that are kind of like a slightly lighter pink color, or depending on how your color vision, maybe they don't look pink, but they're slightly lighter. They're slightly lighter than the surrounding color. These are exons, okay? Exons are expressed regions of a gene. So I'm gonna zoom in on that, maybe, possibly. Oh, no, I'm not. It's just gonna completely go haywire on me. Okay. So I'm gonna zoom in on that so you can see those words. Exons are expressed regions of a gene. That's where the name comes from. The EX from expressed and the ONS from regions. Expressed regions. Cool? So another way that I remember this is that exons will exit the nucleus. So exons they encode mRNA that exits the nucleus. Okay, so that mRNA has to exit the nucleus in order to be expressed, in order to be turned into a protein. So exons encode mRNA that exits the nucleus. Exon is the expressed region of a gene. Another part are the introns. Okay, so these larger sections are called introns. And introns, they get their name from its intervening regions of a gene. Okay, intervening regions of a gene. And so the way I remember, another way to remember this is the mRNA encoded by introns stays inside, so in the nucleus. And we will, don't you worry, we will get into the details of that. Okay. Okay, so we have these elements, these parts of a gene, exons, introns. So if we take our gene, this gene is transcribed into RNA, right? And then that RNA is translated into protein. But when we explain that in that sort of simplified way, which is fine, but now we're getting into more details, right? What we need to understand is that gene is first translated into something called pre-mRNA. And that pre-mRNA is the entire sequence. You can see it's the whole big long length of that gene. But the mRNA is like this little tiny short piece. What is that about? The reason the mRNA is that little tiny short piece is because only the regions of the pre-mRNA that correspond to the exons on the DNA, okay, only the sections of the pre-mRNA that correspond to the exons on the DNA become mRNA. All the other parts get cut out. So they stay inside the nucleus. Then the exons, the parts encoded by exons, are what actually exit the nucleus and what are actually expressed. Okay? So that's what introns and exons are. Okay? Little preview of things we're going to get into in more detail very soon. Okay, so this wide view where we're looking at the entire length of this gene helps us see some important things about genes, that there are introns and there are exons. But we're going to zoom in a little bit further. So we're going to zoom in to just this little section, this little sort of green box. And this little section, we zoomed way in. This little section is only 23 base pairs long. And so remember, base pairs are the base on one side of the DNA double helix paired with the base on the other side of the DNA double helix. So we've zoomed in, we're looking at these 23 base pairs, and this slide, this image shows you both sides of that double helix. So you can see both of the base pairs. But the thing is, any gene is actually only encoded by one side of that double helix. So what I've done is I've removed the other side. So for the purposes of this image, I just oriented the DNA so that the top strand happens to be our gene, happens to be the one we're interested in. So what I've done is I've removed the other side of the double helix to emphasize that point, that any gene is only encoded by one side of the double helix. I'm going to write that down. Okay, so that's a gene, any one gene. So this particular insulin receptor gene is only encoded by one side of that double helix. So this gene, okay? That doesn't mean all genes. Okay, so if we zoom back out, so we zoomed way in so we could see the base pairs. Now, if we zoom back out, what I'm showing you is that on the strand of DNA that we've been focusing on here is our insulin receptor gene, our buddy, our friend that's helping us understand what a gene is. So this is on one strand. So this pink and then the white extending out, this is one strand of the double helix. Okay, and then what I'm showing you is on the other strand of the double helix, make that a different color. So this is the other strand. And over here on the other strand is another gene. So that gene has an awesome, easily pronounceable name. Just kidding, it doesn't. So I'm not going to tell you its name because I don't even know how to pronounce it. But this is another gene, okay? So we have our gene, our insulin receptor gene on one strand, and then on the other side of the helix, we have another gene. So genes are encoded by both sides of the double helix, okay? But any one gene is only encoded by one side. Okay, so you won't see part of the code for the insulin receptor on the top strand and another part on the bottom strand. Okay, but you will see the insulin receptor gene on the top strand and then the RG18 on the other strand. Another important thing to know is that this is actually how these genes are oriented. This is true to life, so that's why I've oriented them that way. But you can absolutely have genes that overlap on the two strands. So you could have a gene here and then another one here. So they can definitely overlap, okay? The thing to keep in mind, and we're going to get into this in a lot more detail, is that genes that are on opposite strands, remember that these strands are anti-parallel. So their nucleotides run in opposite directions. And so they'll be read by the cellular machinery when they're transcribed in opposite directions. And we'll come back to that. Okay, so what's next? All right, so then if we zoom out further, so this is my my just sort of cartoon depiction. If we zoom all the way back out to that entire 58 million base pair long molecule of DNA, remember I told you that is one single molecule of DNA. We zoom all the way out. So now I'm going to tell you that this particular molecule of DNA is actually your 19th chromosome. So your 19th chromosome is a single molecule of DNA. It's around 58 million base pairs long. So this is just sort of my depiction to help you understand what we're talking about. If we look at a true image, so this image on the bottom is taken from NCBI, which is a genomics repository, genomics research. This is a real image of the genes that are really on your chromosome 19. And I've given you the website down at the bottom. Please feel free to go and play and explore if you are at all interested. We're going to be doing lots of exploration into genomes. We're going to be using fly genomes. But so what this is showing you is in the entire length of the human chromosome 19, these green areas indicate where genes are located. Okay, so this is our entire chromosome 19. And so far we know of 2,494 genes that are present on the human chromosome 19. So that's an introduction to the gene. And next we're going to talk about DNA packaging and how these genes are